Hello, it's Pinky from Nepal. Today in this video, I am going to discuss the methods used in the identification of fishes. Since long time and yet now in most of the countries like Nepal, India, Pakistan, Bangladesh and so many other countries, this method has been used for the identification of fish species. The method is known as biometric method. In biometric method, we do two works. The first one is we do meristic counting of the fishes, and the next one is we take the morphometric measurement. Now let us know what is this meristic count. Meristic counting is nothing, it's just the process of counting rays present in different fins of the fishes. So at the time of doing meristic counting of the fishes, in all these fins like dorsal fin, caudal fin, anal fin, ventral fin, pectoral fin, in all of these fishes we count the number of rays and note it. Sometimes what happens, um, in the fins, like here we see in the diagram, in the dorsal fin there are not just the presence of rays. Besides rays, we have also the sharp type of material what is known as spines. So when there is the presence of spines in the fins, we also make the counting of spines. We also do the counting of spines as well as the rays present in the fins. So at the time of counting rays and spines in the fins, it is essential for us to have the basic knowledge what type of structures are called spines and what type of structures are called rays. Sometimes there is a great confusion in between spines and rays. So before counting or doing the work of counting the rays and spines, it is a must for us to have a basic knowledge about the types of spines and types of rays. So this is all about the meristic count which we do for the identification of the fish species. The next one we call is morphometric measurement. Morphometric measurement is nothing, it's just the measurement of the different lengths of the body parts of the fishes as shown in the diagram here. Um, as we see here, different lengths are given, but it's not clear in the diagram. I'd like to proceed in the next fish. Actually, to show the lengths, it's better to take the original specimen. However, I'll try my best to show you how the lengths are taken for taking the morphometric measurement. So here firstly we take the total length which is represented by TL in the chart here we see. So when we take the total length, we take the length between the mouth up to the tip of the fish called total length. After that we take the standard length represented by SL in the chart. Standard length is nothing, it is the length taken between the snout of the fish and the caudal fin base. The next we have is snout length, here indicated by the arrow in the chart. Snout length is the length between the snout of the fish and in front of the eye orbit. It's not clear here. The length between the eye orbit, and the length between the eye orbit and the snout of the mouth is known as snout length. Next comes the eye diameter represented by ED. It is the distance between the two circumferential edges of the eye orbit, means from this part to this part. Next comes the gape of the mouth. Gape of the mouth is actually taken from the ventral region of the head of the fish. It is actually, we say, the distance between the two edges of the mouth. The next comes cleft of the mouth. Cleft of the mouth means, like here it's not distinct from this region to this region. We take sideways, means the length of the, let's say, the jaws of the mouth. It becomes clear when we take the specimen, but it can be still made cleared. It can be still made clear. I'll recommend some few books for that. Let us proceed further. The next one is the interorbital width. It is the distance between the two orbits, and the length taken for it is known as interorbital width, represented by IW. The next one is the predorsal length, represented by PD in the chart. Predorsal length is the length between the snout, up to the front of the dorsal fin, first dorsal fin. The next comes the postdorsal length. Postdorsal length means the length after the first dorsal fin up to the caudal peduncle region. Caudal fin base. From the posterior part of the first dorsal fin up to the caudal fin base. The next we have is the length of the head. The length of the head means from the tip of the snout up to the occiput region. This region is called length of the head. This length is taken. The next we have width of the head. Width of the head means from this part of the head to the next edge of the head. The length is taken ventrally. The next we have depth of the head. Depth of the head means right from the occiput region vertically down up to the base of the head. This length is known as depth of the head. The next we have depth of the body. Depth of the body is taken from the most highest point of the body, from the dorsal up to the ventral part vertically. Length of the dorsal fin we take this way, like if it's a dorsal fin, from the base of the dorsal fin to the tip of the dorsal fin, this is the length of the dorsal fin. Likewise, we have length of the pectoral fin, from the base of the pectoral fin up to the tip of the pectoral fin, this is the length of pectoral fin. Length of the ventral fin we have. So here the ventral fin is not seen. Anyway, from the base of the ventral fin up to its tip, it is the length of the ventral fin. Likewise, we have length of anal fin. It's also the same. From the base of the anal fin up to the tip of the anal fin is called the length of the anal fin. We have length of the caudal fin, means from the base of the caudal fin to tip of the caudal fin, it is the length of the caudal fin. Likewise, we have length of the caudal peduncle. It is the distance, actually the distance from the posterior region of the anal fin up to the caudal fin base. Height of the caudal peduncle means in this region from the tip to the bottom we take the length vertically. It is known as height of the caudal peduncle. Similarly, we take cursor pointer. Similarly, we take the distance between the ventral fin and anal fin. We take the distance between the vent and the anal fin. If there is presence of disc, we take the length of the disc. We measure the length of the disc present in most of the fishes. we take the width of the disc, and we also take the length of the occipital process, which is mostly present in this face called aspirata or it's not still distinct. But when we see in the dorsal region of the head, there is a U-shaped structure which is called occipital process. So likewise, after taking all these various lengths of the face, and after counting the rays and spines present in this face, now we are capable to identify the face. After this, what we do is, after taking these measurements, uh, we move to the website called www.facebase.org, and we find the lens, the morphometric lens, and the meristic count of our face, and compare in this face base. If we get the correct measurements comparing to the measurements or data present in this website along with other external features and characters, then we are able to identify our face spaces. This much for today. Hope the video must have been helpful, mostly with the point of view of the methods used in identifying the face spaces, which was mostly focused on how to take the counts of the rays and spines of the face and how to take the measurements of various body parts of the face. Hope you must have enjoyed the video. Thank you and bye-bye.